today on how I'm going to talk about a different kind of data altogether. So the buzzword of the last decade, that is translational research, is now replaced with the word which is, or the phrase, which is research for public good. Because translational research is actually research which will take the outcome of the research across the valley of death to the communities for their good. So for today we have gathered here to discuss that the health good of the public. So to take care of someone's health when one person is sick, it involves three major steps. One is the detection of the disease, which will lead to the treatment, and then finally the follow-up till the person is absolutely healthy or normal. So all these steps, it emanates a lot of data which when utilized properly can lead to improved products, which we learned since morning till now, which will improve the accessibility of these kind of products and which can change even the practice guidelines. So we know that in the area of detection, we can have devices where AI improved devices, we can have algorithms. Similarly, even for the treatment area, we can have therapeutic devices, we can have diagnostic devices. Diagnostic is the diagnostics which will help in direct treatment, which will improve the treatment. And there are algorithms which will directly help the treatments. Then for today's AI-driven healthcare, databases are parts and parcels of the research or the outcomes now. I'm not going to talk about either of them. So what am I going to talk about? I'm going to talk about different data which will make me design a diagnostic molecule, a diagnostic material, or a drug molecule. Also, I would talk about that these designs, utilization of those data, how it will make me, how it will allow me to make library of molecules. I'm specifically going to talk about a very complex molecule. I'm getting into a little more technical details now. So this is a molecule which is called an antibody. All of you know the name. So this is a very popular molecule with the diagnostic manufacturers or the therapeutic manufacturers. Incidentally, it's also a very popular molecule with the Nobel committees. Being in September, end of September, everyone talks about, the academicians talk about Nobel Prize, so I need to talk about Nobel Prize now. So, because this is a molecule in a different form, it has got 8 Nobel Prizes so far. So this is a molecule which is 150 kilodalton. That means it's a very large molecule, very large protein, which acts as a dimer. That means that the same copy of the molecule, when two of the molecules come together, it forms the active molecule. The green part is the, the green chains here, that is called the light chain. The blue chain here is called the heavy chain. And the light green area is what is called a variable area, that variable region. That means that each antibody's variable region is unique. That's what maintains the uniqueness of the antibody. Whereas that means the rest of the dark areas are the constant regions. That means that irrespective of caste, creed, or religion, everyone in the same species will have same, or I would say the similar sequence of the antibody. With this blue stem, this is a Y-shaped structure, right? With the blue stem, as you would see here, that this is attached to the B cells where it is formed and till it gets secreted. Once it gets secreted and comes to the pathogens or the antigens to which it will bind, it binds through the tip of the Y, it binds to the pathogens and acts on it. If it is only the finger-like structure at the end of the arms, then why is the antibody so large? This is another view of the antibody. The black part is the FC region, that is the stem, and the red parts are the arms which binds to the antibody. I don't expect you to read this busy slide or even to remember this. I just want to convey the message that the antibody is very large because it is not only the antigen binding regions which are important, every part of the antibody that has a function. So every part of the antibody is required. So when a pathogen enters our body, after that, typically we get a million copy of the antibody. So such a complex molecule which is a very favorite molecule of the diagnostic manufacturers or the therapeutic manufacturers. Now, when I said that we get a million copies of the antibody or a million different kinds of antibody, does it mean that we have million genes for that? No. What we have is that we have just wanted to tell you about the complexity of the molecule, that it has the antibody genes actually come from 3 different chromosomes. The 2 types of light chain which can come from 2 chromosomes, chromosome 2 and chromosome 22. The other one can come from, the heavy chain comes from chromosome 14. Now all these different, the red ones, they form those genes, those are all individual genes. So they form the variable region, and which I mentioned in the previous slide. But it is not enough that all the variability comes only from the variable region. Something which is called the J region, the joining region, which forms the antibody, that also leads to the diversity. And the heavy chains are not satisfied only with these 2 diversity. They have a specific set of genes which are called the diversity genes. So all these, the B, the J and the D regions, they form the antibody, they bring in the diversity. To that added are the constant regions, and the constant regions, the number of the numbers are written on top. So the variable region genes can vary from 29 to 46, whereas the J chains are a little less variable from 1 to 6. And the constant region, the heavy chain has maximum number of constant regions, and that can have 9 different kinds of constant regions. Different constant regions are put into the antibody as the immune response starts. So that means that first, as soon as the pathogen enters, we get the C new kind of constant region, which gives rise to what is called IgM, which is a very large antibody, so on and so forth. So this leads to the variation. So that means in a nutshell, in a large nutshell, so we have around 200 antibody genes which can give rise to 10 to the power 18 million possible antibody sequences. So here we have huge data. Here we have large data, very large data. So with this now, just a quick view to how these antibodies are formed. So as soon as the pathogen gets into our body, the genes, the DNA, our DNA that twists and turns, and at the DNA level, it forms this V, D, J regions. They combine and they form the, right now, through a process called somatic recombination, it gives rise to a rearranged DNA, which is transcribed to the pre-mRNA, which again gets cut and pasted in random to give rise to the mRNA, which translates to the polypeptide, 2 polypeptides, the heavy chain and the light chain, which then combines to make the antibody. such is the very complex process of making the antibody. No doubt when we have infection, our body is heated up. Talking about it and getting fever. So, sorry. So just to say that this VDJ recombination, it won the Nobel Prize in 1987. Not the first Nobel Prize, not the last Nobel Prize, but this is so important. Now, how do we use this in making a diagnostic? Now, making the antibody, the first thing is that antibody development, that's the material development. Antibody development that happens in an animal, or from the animal, the antibody producing B cells are taken, and that has been immortalized, that is grown in that's grown in a cell culture dish. And after that, so that gives a large number of antibodies. After that, there is a huge step of screening, when we find out which antibody is the most important antibody, which antibody is the most efficacious one, which will produce the maximum amount of antibody. So, and then the standard steps of prototype development, validation in patient samples, then to get the approval, get to production in the market. Now when we bring in AI here, that means when we bring in design, we can completely remove the step of screening, and so the all other steps of everything, all other steps to make it to a diagnostic, that's research. So antibodies were first used for human benefit around before 1900, and when it was used as vaccine. It took around 3/4 of a century to immortalize the antibody producing B cells and to bring it to cell culture. When we first we could make antibodies which are specific to a target, we could think of using those as a therapeutic antibody. But then two more techniques, two more technologies. One is by Sanger, the DNA sequencing technology. The other is by Mullis and Smith, the PCR amplification technology. So those actually, it could give rise to another huge improvement of the steps, which made the PHAJ library, and that so where we could make a library of recombinant antibody, and that actually exploded the number of therapeutic antibodies. The therapeutic antibodies, once we had the therapeutic antibodies, that became the series of antibody data, which now it has given rise to, thanks to the work of Charlotte Dean and Oxford and Jeff Bray and Johns Hopkins, that we have now the antibody databases, which is a huge database of antibody structure and function relationship, which the size of this database is now around 1 million, and there are multiple AI algorithms which has set the stage for de novo antibody design. So we can completely come out of animals, and we can design antibodies and take them to the market. So are we using it? This is the workflow. This is my last slide. The workflow which we are using now is that we are taking from patient samples, we are taking the PBMCs, we are taking the sera and checking that whether such antibodies exist. And if the antibodies exist, we take out the PBMC, we take the RNA from there, sequence those, and by various bioinformatics analysis, find out that which are the antibodies, and then once we know that this is how we design the antibodies now, and then make it to the to recombinant antibodies, and then test it out by mass spectrometry. The blues, the areas in blues, those are the experimental steps. So we test it out, and that's how the antibodies are made now. So we are closer to making de novo antibodies, and just the kind of projects that we are working on are from infectious diseases, cancer, neurological diseases. different forms of antibodies can be made so that we don't have to use very complex production system. This is how we can decrease the cost of the antibodies, and the final goal is make antibody therapy at the cost of antibiotic so that it can be taken to. So this is being done in the organization CPDTRI, which is a very new organization, and where it's a research and education institute which is for public good. And we formed this institute to walk the last mile across the valley of death in translational research. I don't have to mention this. I just want to thank Pamela Madam and Dr. Kundu, who started this trust, and thank my computational biology collaborator, Dr. Sujan, who is doing the hard work for doing this, and thank Dr. Devnath for agreeing to collaborate. And we've started this journey now.